Welcome back to AAT Learning Hub. I'm Bhumika Anand, and today I'm going to talk about the most abundant cell in our body. My last video was on hematopoiesis. So as a quick reference, hematopoiesis is the process by which the body produces blood cells, also known as the formed elements of blood. And what are they? Yes, the RBCs, WBCs, and platelets. So all the cells develop from a primitive stem cell which is pluripotent, meaning it can differentiate into any kind of cell as well as retain its original stem cell abilities. So this pluripotent stem cell is the hematopoietic stem cell, which differentiates into a myeloid progenitor cell and a lymphoid progenitor cell. The myeloid progenitor cell then undergoes several divisions to form mature erythrocytes, monocytes, granulocytes, which are the neutrophils, eosinophils, and basophils, and thrombocytes, while the lymphoid progenitor cell differentiates to form the lymphocytes. So today we are going to focus on the most abundant cell, which is the erythrocytes. Erythropoiesis is the process which involves the origin, development, and maturation of erythrocytes. So where does this occur? In fetal life, it initially occurs in the yolk sac, which is the mesoblastic stage. Then from the second month in the liver, spleen, and the lymph nodes, which is the hepatic stage. The final or myeloid period begins in the fifth month, with liver occupied with erythropoiesis and bone marrow with leukopoiesis. Later, bone marrow takes charge of overall phemopoietic activity. So after birth, up to 6 years, all bones are filled with red bone marrow. Hence, RBC production happens in all the bones. By 20 years, almost all the long bones are replaced by yellow bone marrow. So RBC production in these locations will stop. By adulthood, only the vertebrae, the ribs, the flat bones, and the upper ends of femur and humerus produce red blood cells. Let's see the stages of development. So first you have the hematopoietic stem cell, which differentiates into a common myeloid progenitor cell. So this common myeloid progenitor cell will differentiate into a proerythroblast, which is the beginning of RBC production. Proerythroblast is about 14 to 19 micron in diameter. They have a large nucleus with nucleoli and reticulum of fine chromatin threads, but they do not contain hemoglobin. The next stage is early erythroblast, where the cell becomes 11 to 17 micron in diameter. The nucleus is still present, but nucleoli is absent. This stage is also called basophilic erythroblast because when examined under the microscope, it takes up basic dyes on staining. The next stage is late erythroblast. This is a very important stage and is also called as polychromatic erythroblast because this is when the hemoglobin starts appearing. The nucleus chromatin starts condensing to accommodate hemoglobin. Then comes the normoblast stage, wherein the cell is about 7 to 10 micron in diameter. This stage is called orthochromatic because of the concentration of hemoglobin in this. Hemoglobin starts increasing in this stage. The nucleus shrinks and looks like an ink pot. This process is called pycnosis, and this stage is the last stage before the nucleus gets extruded from the cell. The next one is the reticulocyte, which is also an immature red blood cell. In this stage, the nucleus is pushed out of the cell and the cytoplasm contains reticulum, which is why it is called reticulocyte. The last one is the erythrocyte, which is the mature RBC. Here, the ribosomes are also shed off and it doesn't contain any nucleus. It takes about 7 days' time to pass from the proerythroblast to that of the reticulocyte stage. The reticulocytes enter the bloodstream and it takes another 2 days to form mature RBCs. So normal red blood count. Normal RBC count in healthy adults is about 4.5 to 5 million in females and 5 to 6 million In males, per milliliter of blood. In infants, it's about 6 to 7 million. In the first 10 days of postnatal life, large number of RBCs are destroyed. This is one cause of jaundice in newborns. So the stages of development of RBC is very easy. Initially, the cell sizes are big and they start decreasing. Hemoglobin, which is not present initially, starts appearing and increases. Nucleus and nucleoli that are initially present, they start condensing, and finally the nucleus is pushed out. So a mature RBC is small. They do not contain any ribosomes, do not contain nucleus, and is full of hemoglobin. Let's look at the shape and structure of erythrocytes. This is very interesting because it's biconcave shaped. Concave means that which curves inwards. And it's interesting because the RBCs have this shape on both the sides. Hence it's called biconcave. They are plump at the periphery and very thin in the center. The discocyte shape of human RBCs is approximately 7.5 to 8.7 micrometer in diameter and 1.7 to 2.2 micrometer in thickness. When erythrocytes enter the capillary beds, they move very slow as the capillaries are extremely narrow, and this helps in gas exchange to occur at a great extent. But how can RBCs pass through such small capillaries? Fortunately, their structural protein called spectrin is flexible, and this allows them to bend or fold over themselves to a great extent to travel while in capillaries, and then they spring back again when they enter a wider vessel. Next, the red blood cell does not have mitochondria and other organelles which is otherwise present in a normal cell. What is the main purpose of mitochondria? Is to create energy through respiration, which requires oxygen. But when RBC's primary role is to transport oxygen, do you think it's fair to utilize the oxygen? It's like your food delivery partner eating up a portion of the food that he's supposed to deliver to you. So here, our RBCs are very professional. They do not utilize any of the oxygen they are transporting, so they can deliver it all to the tissues. Hence, RBCs lack mitochondria and depend on anaerobic respiration. Finally, RBC, they do not have nucleus, so they have more space to hold oxygen. Also, the structure of hemoglobins, which is made up of 4 ion known as heme and proteins known as globins, helps the red blood cell to carry more oxygen than it otherwise can. In the bloodstream, iron picks up oxygen in the lungs and drops it off in the tissues. The amino acids in hemoglobin then transport carbon dioxide from the tissues back to the lungs. The mature RBCs have a lifespan of up to 120 days, after which they are expelled out of the bloodstream and replaced by new RBCs. If there are too many RBCs, the blood will thicken, impeding or slowing the flow and cause blood clots. And if RBC count is low, your body will not receive sufficient oxygen. So there must be something that can regulate the right amount of RBC in our body, correct? And this is where our kidneys come into action. Erythropoietin or EPO is a hormone produced primarily by the kidneys with small amounts made by the liver and the brain. Whenever the kidneys sense a low blood oxygen level, which is hypoxemia, it produces erythropoietin and releases it into the blood. The amount of erythropoietin released depends on how low the oxygen level is and the ability of the kidneys to produce erythropoietin. So this EPO is carried to the bone marrow where it stimulates production of red blood cells. Now, as oxygen levels rise in the blood, the kidneys will detect an increase in hemoglobin and will slow down the production of EPO. This way, your body achieves a balance, homeostasis, where you have the right amount of red blood cells. If the kidneys are damaged and do not produce enough erythropoietin, the RBC production also decreases and you can become anemic. Similarly, if your bone marrow is unable to respond to stimulation from EPO, then again you become anemic. Higher levels of EPO may be seen in people who live at high altitudes or people with lung disease because the kidneys produce more EPO to try to compensate for the low oxygen level. Likewise, if there is too much of erythropoietin, RBC production will also increase, and this will lead to polycythemia or erythrocytosis. There is also a condition called polycythemia vera, which is caused by a bone marrow disorder, and it is not primarily because of increased erythropoietin. So apart from erythropoietin, there are other factors which are necessary for development and maturation of RBC. A quick look at them. General factors, as I told you before, is erythropoietin. Then you have the thyroxine, the T4. This hormone accelerates process of erythropoiesis. Hematopoietic growth factors like the interleukin 3, interleukin 6 and interleukin 11, and vitamins like vitamin B, C, D and E. For maturation, you require vitamin B12, which is essential for synthesis of DNA in RBC. Intrinsic factor. Intrinsic factor of Castle is produced in gastric mucosa and is essential for the absorption of vitamin B12 from the intestine. Folic acid is essential for synthesis of DNA. Factors that are necessary for hemoglobin formation. First-class proteins and amino acids for the synthesis of protein part of hemoglobin, which is the globin. For the heme part, you require iron. Copper for absorption of iron from GI tract, cobalt and nickel for utilization of iron, vitamins like vitamin C, riboflavin, nicotinic acid, and pyridoxine are also essential. Now, the lifespan of RBC is 120 days. So what happens to the red cells after their lifespan? The phagocytic cell called macrophage engulfs the aged or damaged RBCs. This phagocytic activity occurs predominantly in the spleen but can also occur in the liver and bone marrow. Hemoglobin in the RBC is broken down into globin and heme. Globin is further broken down into amino acids which enters the circulation and is sent back to the bone marrow to be used in the production of new erythrocytes. The heme portion is broken down into bilirubin and iron. The iron, with the help of a transport protein called transferrin, through the liver enters bone marrow for erythropoiesis. It also gets stored in the liver in the form of ferritin or hemosiderin. Bilirubin binds to albumin and travels in the blood to the liver, which uses it in the manufacture of bile. In the intestine, bacteria breaks the bilirubin and converts it to urobilinogen and then to stercobilin. So this is all about erythropoiesis. I hope this video was helpful. Stay connected with AAT Learning Hub. Thank you and happy learning.